In this video, I am going to talk about Hybridoma technology. Each hybridoma constitutively express a large amount of one specific monoclonal antibody, and favored hybridoma cell lines can be cryopreserved for long-lasting monoclonal antibody production. As a result, researchers usually prefer generating hybridomas over other monoclonal antibody production methods in order to maintain a convenient, never-ending supply of important monoclonal antibodies. Now, what is hybridoma technology? Hybridoma technology is a well-established method to produce monoclonal antibodies specific to antigen of interest. Hybridoma cell lines are formed via fusion between a short-lived antibody-producing B cell and an immortal myeloma cell. Before the mid 1970s, researchers using polyclonal antibody were met with many challenges, such as the need for or repeat use of large numbers of immunized animals to acquire them due to the lack of immortalized antibody producing cell. Then the high levels of the background and lot to lot inconsistency of experiments that came from the broad specificity afforded by these many antibodies for single antigen with different epitopes. Hence, a key issue of the time was inability to isolate and purify single antibodies of singular known specificity of monoclonal antibody from the many different kinds of product produced in the body. Due to lack of specificity and heterogenic nature, there are several limitations on the utility of polyclonal antibodies for the therapeutic and diagnostic purpose. In the early years, animals were immunized against a specific antigen. B lymphocytes were isolated and cultured in vitro for producing monoclonal antibodies. This approach was not successful since culturing normal B lymphocyte is difficult and the synthesis of monoclonal antibody was short-lived and very limited. It means the lifespan of B cell is very short. It is interesting that immortal monoclonal antibody producing cells do exist in nature. They are found in the patients suffering from disease called multiple myeloma. It's a cancer of lymphocytes. Hybridoma technology was discovered in 1975 by two scientists, George Kohler and Cesar Milstein. They wanted to create immortal hybrid cell by fusing normal B cells from immunized mice with their myeloma cell. Their discovery is considered one of the greatest breakthroughs in the field of biotechnology. Because for the past decades, hybridomas have fueled the discovery and the production of antibodies for the multiple of application. Here the antibodies are monoclonal antibodies. And the result is that the artificially immortalized B lymphocytes can multiply indefinitely in vitro and produce monoclonal antibodies. The hybridoma cell possesses the growth and multiplying properties of myeloma cells but secrete antibody of B lymphocyte. The production of monoclonal antibodies by the hybridoma cells is referred to as hybridoma technology. The following steps are involved in
Hybridoma.
myeloma technology are as follows. One is immunizations. Next step is cell fusion. Next one, selection of hybridomas. Next, skinning the products. After that, cloning and propagation. And last is characterizations and storage. The very first step is hybridoma technology is to immunize a animal, usually mice, with appropriate antigen. This enables increased stimulation of B lymphocytes which are responding to the antigen, to the foreign antigen. The immunostimulated cells for synthesis of antibodies have grown maximally by this approach. And when the serum concentration of antibodies is optimal, the animal is sacrificed. And then the spleen is aseptically removed and disrupted by the mechanical or enzymatic method to release the cell. The next step is cell fusion. The thoroughly washed lymphocytes are mixed with HGPRT defective myeloma cell. This HGPRT means hypoxanthine guanine phosphoribosyl transferase. This HGPRT defective myeloma cells and the mixture of cells is exposed to polyethylene glycol for a short period. Since it is toxic, polyethylene glycol is removed by washing and the cells are kept in fresh medium. These cells are composed of a mixture of hybridomas that are fused cells, free myeloma cells and the free lymphocytes. And the third step is selection of hybridomas. When the cells are cultured in HAT medium, HAT means hypoxanthine aminoterine and thymidine. HAT medium only the hybridoma cells go while the rest will slowly disappear. And the next step is the skinning the products. The hybridomas must be skinned for the secretion of antibodies of desired specificity. The culture medium from each hybridoma culture is periodically tested for the desired antibody specificity. Two techniques, namely ELISA and RIA, are commonly used for this purpose. In both the assay, the antibody binds to the specific antigen, usually coated to plastic plates, and the unbound antibody and other components of the medium can be washed off. Thus, the hybridoma cell producing the desired antibody can be identified by scanning. The antibody secreted by the hybrid cell is referred to as monoclonal antibody. And the next step is cloning and propagation. The single hybrid cells producing the desired antibody are isolated and cloned. Two techniques are commonly employed for cloning hybrid cell. One, limiting dilution method. Another one is soft agar method. And the last step of hybridoma technology is characterizations and storage. The monoclonal antibody has to be subjected to biochemical and biophysical characterization for the desired specificity. If you look for the overall technology of hybridoma, the very first step is immunizations, where the immunized animal, usually a mice, immunized with an appropriate antigen, and this enable increase the stimulation of B lymphocyte. which present in plasma cell, and it's which are responding to the antigen, to the foreign antigen, and immune stimulated cell for synthesis of antibody have grown maximally by this approach. And this is thoroughly washed lymphocyte are mixed with HGPRT defective myeloma cell or tumor cell or cancerous cell. And this B cell, the characteristics of B cells are it has short lifespan, it has HGPRT enzymes, and the myeloma cell, the characteristics of myeloma cells are it don't have any HGPRT enzymes, and its lifespan is immortal in nature. Okay, and if you fused the both of B cells and myeloma cells, you get the 5 types of cells. One is fuse plasma, fuse myeloma. Unfused myeloma, unfused plasma. And hybridoma. This is hybridoma cell. The myeloma cell used in hybridoma technology must not be capable of synthesizing their antibodies, own antibodies. The selection of hybridoma cell is based on the inhibiting nucleotide or consequently DNA synthesizing machinery. The mammal cell can synthesize nucleotide by 2 pathways. One is de novo synthesis and another one is salvage pathway. The formation of tetrahydrofolate formed from dihydrofolate, and the formation of the tetrahydrofolate, therefore the nucleotide can be blocked by inhibitor aminotridine. The salvage pathway involves the direct conversion of purines and pyrimidines into the corresponding nucleotides. Hypoxanthine guanine phosphoribosyl transferase, which is HGPRT, is the key enzyme in the salvage pathway of purines, and it converts hypoxanthine and guanine respectively to the inosine monophosphate and guanosine monophosphate. And the thymidine kinase is involved in the salvage pathway. This thymidine kinase is involved in the salvage pathway of pyrimidine, converts thymidine to thymidine monophosphate, and any mutation in either one of the enzyme like HGPRT and TK, which is thymidine kinase, blocks the salvage pathway. When cells deficient, these all are mutated cells in HGPRT are grown in medium containing het medium, which is hypoxanthine, aminotridine and thymidine. They cannot survive due to inhibition of de novo synthesis of purine, because in the het medium their aminotridine is present, and aminotridine is inhibitor of the key enzyme of de novo synthesis, which is dihydrofolate reductase. So there they cannot survive due to the inhibitions of the de novo synthesis of purine nucleotides and salvage pathway is not operative because lack of HGPRT, because plasma cells have HGPRT. Myeloma cells don't. Here also, myeloma cells don't have HGPRT and plasma cells have HGPRT. This is fused plasma, this is fused myeloma, this is unfused myeloma, and this is unfused plasma, and this is the hybridoma. But in the absence of HGPRT, the myeloma cells cannot survive in the het medium. But in the presence of HGPRT, presence in both fused and unfused plasma cell can survive in het medium for few days, because the lifespan of plasma cell is very short. And in the case of hybridoma cell, it can survive and can produce a monoclonal antibody because the presence of HGPRT and immortal in nature, because hybridoma cells are formed from one B lymphocytes which comes from B cells, and another these B cells fused with immortal nature cancerous cells or myeloma cell. So it contains both of HGPRT and immortal in nature, so it can survive in het medium. And some advantage of monoclonal antibodies. Those are monoclonal antibodies truly represent a homogeneous state of a single molecular species, and each specific monoclonal antibody is specific to a given antigenic determinant. And it has some limitations. There as follows: Hybridoma's technologies is laborious and time consuming, and they cannot differentiate the molecule as a whole. Monoclonal antibody are produced against a single antigenic determinant, and the process are great danger, because since there is no guarantee, the monoclonal antibody produces totally virus free despite the purification, but it has lots of application. This hybridoma technology has great important value in biological purpose, like pharmaceuticals purpose, like biotechnological purpose, biophysical purpose. Hope this video will help you to understand the hybridoma technology and the steps are involved in the hybridoma technology, and the mechanism are involved in the het medium, and the advantage of monoclonal antibody, and what's the disadvantage of polyclonal antibody, and the limitation of the monoclonal antibody. If you like this video, please share with your friends and subscribe, and don't forget to subscribe, and don't forget to press the bell icon to get more video relating the biological topics which help you in your study. Thank you.